Lunch with the Core. I'm Dr. Glenn Freed, and so I'm presenting an introduction to the Core today. This is an ongoing lunch series that we have in the spring and the fall for about maybe 9 or 10 weeks each semester, and it's a way to let people get acquainted with other people doing research in the Core, gives people an update on what's going on. Typically, it's research projects that are at least partially taking place in the Core. And then we do a few other things like instrument demos and discussions from Core staff. So go ahead and grab some pizza and we'll get started. So kind of an overview of the goal of the Core is to provide staff and instrumentation to help researchers produce scientific data. And so, I mean, this is pretty clear. Graduate students and postdocs and a few undergrads come and work with the Core staff. We have instrumentation that we maintain, and they can come in and use it. They learn how to use it, and so on. So the most important part in Core facilities is the staff, and so this is our people. Kingsley and Austin have been working with us for about 8 years now. They do training. Kingsley does a lot of the serial block face and MinFlux work. Austin does a lot of training on the other instrumentation. Duncan and Umina both joined the Core this spring and early summer. And they are doing training on the confocals, wide field microscope, the slide scanners, and things like that. Hannah takes care of billing, and so if you have a question with your CFOP or something like that, you can talk to Hannah. Hannah also sets up the pizzas, and so you can tell Hannah thank you today when you go to leave. Miranda is sitting over there, and she takes care of training for the NanoZoomer and the Axioscan and does lab safety and kind of takes care of the lab. So other things going on, we do conferences, like we did a MinFlux conference. We bought a new LSM 900 in January. So those are the kind of things that go on during the year. We do a lot of training, and so that means that if you want to use the Core, the standard operating procedure is to go to our web page, fill out a training request form. I usually do a basic optics class that covers optics and how you work in the Core every other Friday. And then we do hands-on training. We expect that to cover a small amount of the information you should learn in the Core. The other part you should pick up by asking questions to staff people as you progress through your research project. So each project is a little different. You want to know exactly, how to use the microscopes, what's going on. If you have questions when you start collecting data, the staff people are a good resource. Occasionally we do kind of special projects that are a little bit beyond that. And so this is our image of the month this week, or this month. This is from Andrew Leakey's group, and it's Joe Crawford collected it. And so the story behind this project is that a few years ago, they were interested in imaging stomata opening and closing. And so that's a little bit difficult because you have to control the environment around the leaf, and you also have to image it. So it became clear we had to modify our LSM 710 The core staff suggested an objective to use, built a little adapter, built a stage adapter for their instrument to supply CO2 and measure the gas environment, and now they can monitor the gas environment around the leaf, and they can watch these stomata open and close in time. And hopefully in the spring, Joe will be presenting about that. One other thing I should let you know under the staff category, we're probably going to start looking for a postdoc in the next few weeks. And so if you know someone who really likes working with instruments, really likes doing research and thinking up ideas on how to solve research questions, that would be a great position for them. The cool thing about being a core staff is you get to meet somebody with a really cool research problem on Monday morning, and you can talk to them about it and set up a microscope to help them solve the problem. And then on Monday afternoon, you can do the same thing. Tuesday morning, you can start another cool project, and you can start a fourth cool project Tuesday afternoon. And you get to do that for years. And so it's just a lot of fun. Okay, so we'll keep going. This is kind of a picture of the size of the core. We work with 127 research groups from about 30 different departments all across campus. So up north, the engineers, a lot of engineers are looking at cells, and sometimes we work with other mechanical structures, and then you can see this whole list all the way across the departments. The other key thing that people should ask when they're looking at a core facility in general is how busy are your instruments? Because if you're using a core where the instruments are booked all day long, good luck getting any work done, right? And so a normal work year is about 2,000 hours. And so you can see that our instruments are well used, but they're not used 8 to 5, for example. And with our calendaring system, you can come in 24 hours a day, 7 days a week. So while they're probably, like our confocals are currently busy most afternoons, if you want to come very early in the morning or in the evening or on the weekends, there's free time to use all of our instruments. This is also a number that people look at if you write a grant for a new instrument. And so if these numbers are less than 2,000 hours, they're going to say, the next guy has 3,000 hours. You can at least access something. Okay? Another question to ask about a core facility is, are their instruments all aging or not? And so quick list or look at this history here, and you'll see that we have brought in 14 new instruments in the last 10 years. And so we're bringing in about 1.5 instruments a year. There's a few times when we're faster and a few times when we're slower, but overall that's what we're averaging. The other point about this is we got columns on dates, but we also have this support column here, right? And so when we buy instruments, it's not just because Glenn gets an urge to go buy something. It's because we talk to faculty and there's a group of faculty on campus that need a certain technology. Sometimes I see some new technology out there that I think would really apply to specific projects, and I suggest it. But if there's not a user base to actually use it, it would be kind of ridiculous to actually buy it, right? So what we do is we talk to faculty. These are the people that kind of pushed each purchase through, and you can see there's a wide variety of departments and groups here. The MinFlux is a good example, kind of a nice story because, yeah, it's a nice story. So it's about a $2 million instrument, and we got us a little bit of a discount on that. Zan and Martin raised money for this from departments across campus for about half of the money, and then we received a Carver grant for the other half, and so that covered this. At the same time we applied for the Carver grant, we applied for an NSF grant, and so we got a little bit of extra money for that, and so we could buy the Steadicon from that extra money. The rest of this story is the reason they were interested in this was they wanted to show that campus was an ideal place for a science and technology center on quantitative biology. And in that science and technology center, what they wanted to do is collect data on the location of molecules in a cell down to 2 nanometers. And this is the instrument that could do it. And so then that Science and Technology Center was funded, and the announcement came out, I think, last week. And in fact, a little announcement is right outside today. And so I didn't plan that, but that's why that's a cool story. Okay. So we'll start by talking through a bunch of the instruments we have. We'll start with the MinFlux, since I was just talking about it. So here's a picture of our MinFlux with two of Martin's students. In the spring, we hosted the Midwest MinFlux and Super Resolution Conference workshop. We had 120 people register for that. When we bought our MinFlux, we were the 3rd MinFlux in the U.S., and so there's one on the East Coast and one on the West Coast, and so this was a great way to introduce people from both the Midwest and the coast, actually, to the MinFlux technology. We did 13 lectures during 4 days. People like Stefan Hell and Christian gave talks, and also Kingsley also gave a talk, and so we recorded all of those. You can find those on our website by either looking at this code, or you can go to our outreach page and scroll down a little bit, and you'll see a link to all those lectures. And so that's a great way to learn the details about MinFlux and labeling and STED technology if you're interested. Next year, on May 20th to the 24th, we're going to have the 2nd annual Midwest MinFlux and Super Resolution Workshop. So I was going to cover how MinFlux works quickly, so you don't have to, you get a kind of an overview of the lectures. Here's the instrumentation, and then this is kind of the way the beam works. So MinFlux is a fluorescence microscopy technique. So you have a light source, and then there's 3 things that you do to your light source. You have a phase mask that makes a donut, so instead of illuminating the whole sample evenly, you only illuminate this donut shape. You have something to turn the laser intensity up, and you have an XY beam deflector. So that gives you the ability to move your donut around. So then when you're doing MinFlux, you want to get 2 nanometer resolution, you have a bunch of fluorophores in your sample, but you turn these on one at a time. So these are highlighted as turned on, and you turn one of them on, then and then it burns out and goes away. You turn the next one on, then it burns out, then you turn the next one on. So you turn those on with a blue light, a 405 nanometer light, and you cover about an area about 250 nanometers wide of what you turn on. And then your donut is a little bit bigger than that. So this is about 100 times bigger than the resolution. And you start with your donut over where you think your molecule is, which is this star here, and then you move it around a little bit. So you move it up so the center is up here, and when the star is in near the center, you get very few photons. When you move it up here, now your fluorophore is where the excitation is more intense, so you get more photons. Then you move your donut down here, and now you're also getting more photons, but not quite as many maybe. And then you move it over here, and now you're getting a lot of photons, right? So now you have information from actually four different locations, and you can make a guess at where your fluorophore is. In reality, what we do is we use 6 spots to do our search if you're doing 2D, and then you use a search pattern like this if you're doing 3D. And so now your resolution, when you work out the math, the resolution is related to the L on your search pattern, and it's not tied, well, it is tied to the number of photons, but now you can locate things with much fewer photons. And that's important because these fluorophores burn out after about 20,000 photons or something like that. So that's a really quick way to see how you can get 2 nanometer resolution. 2 nanometer resolution is the size of small molecules. And so, in fact, fluorophores that we normally use are drawn up here to scale, and you can see that 2 nanometers is smaller than many fluorophores that most people use in fluorescence microscopy most of the time. The photophysics of the fluorophores are actually very important, and so we typically use Alexa-4 647 when we're doing imaging, and sometimes we use CF 680. Kingsley is a great resource for discussing labeling. If you use both of these and you're going around and you find a fluorescent molecule and you locate it, you may not know which of these you found, right? But you'd really like to know because that gives you the ability to find the location of 2 different objects. So what we have set up in the MinFlux is 2 detectors, and there's a dichroic sitting right here that's right between the emission of the 647 and the 680, and that way we can separate these 2. So now we can do essentially 2-color imaging with our MinFlux. When you want to do tracking, to track like molecular motors, for example, as they're stepping, we would use CF 633 or Alexa 488, we would use the 540 nanometer laser for this, and we'd use the 640 for these other things. Okay, so most optical microscopy cores are proud of their confocals, and we are just the same. We're also proud of our confocals, so we have 4 of those. LSM 700 and 710 are admittedly aging at the moment, but I did a resolution check the other day on the 700 and it looked really good. So you should have confidence that it's not, the quality of your images are not decaying over time here. It's a 2-channel system, and the 700 has 4 laser lines. And so in fact, most biological imaging can happen here. The 710 has a wider selection of CW lines. It has a 70 femtosecond Ti sapphire, so we can do 2 photon and second harmonic generation. It has a spectral detector. It has a fluorescence lifetime detector. It has a high sensitivity detector for transmitted SHG. So it has many, like if there's a paper that you do see some confocal work done, the 710 is probably capable of doing that. We have an LSM 880. Unfortunately, we sent out an email yesterday or the day before that notified people that our LSM 880 had to be returned to Germany for a couple months. So this is where we give everyone a talk about don't spill liquids on your microscope, because then it goes back to Germany. When it comes back, it has the ARIScan capability with 140 nanometer resolution. So the way this works is our the point spread function of our light going through the confocal is spread out over 32 detectors, and then you can measure the shape of the point spread function, and now your resolution is improved. These have about 250 nanometer resolution, and with the ARIScan that these two confocals have, we can get down to about 140 nanometer resolution by measuring the shape of the point spread function. As I mentioned earlier, the LSM 900 was installed in January, and it's heavily used now. It's a 2-channel system, plus it has ARIScan, and the other thing it has is a deconvolution package, which can get down to about 90 nanometer resolution if you're interested in that. Typically, the 250 or 140 nanometer resolution is good enough. It also has a nice sample finder and tiling setup, so if you're interested in tiling, this would be a good confocal to use. If you're a person that has been using the LSM 880 and now you find out that you can't for a month or so, let us know, and we will try to move you to one of these other confocals. Okay. As an effort to add one more confocal plus a STED capability, we are purchasing the SteadyCon system right now. And so this is midway through the purchasing process. It will give you 40 nanometer resolution, so this is much better than the 120 or 140 to 90 nanometers from the LSM 900, and it's significantly worse than the MinFlux, so it's kind of in the middle there. The way STED works, here's our energy diagram, so you excite like you would in a normal fluorescence microscope, but with STED, some of the fluorophores you force down to the ground state with an extra laser, and that's why it's called stimulated emission, because here you stimulate the emission and then you get normal fluorescence in other locations. So that's the energy diagram. This is the spatial diagram. Here you have the excitation light. This is spread over about 250 nanometers. So here's your fluorophores before they're excited. Here you excite a 250 nanometer region. Then you deplete or you stimulate the emission in this donut shape. And so all of these guys out here are forced to the ground state with this path. And then the final step is this path over here is only applies to this single fluorophore here. And so, or the fluorophores that are, depending on your labeling density, the fluorophores that are in the center of your donut. So depending on your intensity of your laser, of your depletion laser, compared to the saturation level, you can now decrease your resolution down to 40 nanometers. And we did, UMNA did the demo on this with several people and could demonstrate the 40 nanometer resolution without a problem. I need to remind, or I need to point out that this is 40 nanometers in X and Y. STED is possible in 3D, but the STED we purchased is a single plane STED. We also have 405, 488, 561, and 640 nanometer lasers that work in confocals. So you can image in confocal mode, and then you can overlay the STED capability, the STED resolution on top of that. Okay, so here's we have here's some more microscopes. We have a super resolution structured illumination microscope. This works by projecting a grid across the surface of your slide and then imaging that grid in different positions. So it can get about 120 nanometer resolution. It has the normal excitation sources. It has an EMCCD camera because now you're imaging the whole slide all at once. Oops, this is like doubled. We also have slide scanners. So these are tools where you can image whole slides all in one shot. We have two of those. We have the Nano Zoomer, which we purchased first, and then the Axioscan. The Nano Zoomer is great for bright field slide scanning. We provide data for the art of science, and so if you're interested in getting some of your data into the art of science, let us or Julia upstairs know. We decided to print out one of our NanoZoomer data sets at 300 dpi, and what we end up with is an image that's 12 feet tall and about 8 feet wide. So that's how much data you have in each image. So this is an image of a mouse embryo that was sectioned and put on a slide. Our Axioscan is good for slide scanning in fluorescence or polarization, so you can do all the slides that you couldn't do in bright field can be done with the Axioscan, and it has the same kind of capabilities. Light sheet microscopy is a way to look at tissues. So now we're kind of stretching our resolution from very small to lower, but our sample sizes are growing. So if you think about something such as a mouse brain or a mouse heart or some organ, you can clear that with our X-Clarity setup. And then you excite with a plane of light going one direction and objective going the other. And so we have two of these systems. The Zeiss Light Sheet Z1 works with smaller tissue samples, and the ultramicroscope works, this is the ultramicroscope here, and it works with larger sections. Okay. We have a Raman AFM setup, so this is from YTech, and we can do both Raman imaging. It's a confocal system, so you can do confocal Raman, and then you can do AFM. There's an AFM setup on one of the objectives here that you can do over the same areas. So for polymer samples, for example, you could do a Raman to identify the polymer present, and then you can do AFM to measure surface adhesion or surface topography. The GARD group uses this a lot to look at concrete samples, and these are spectra from different locations in the image, and that tells which crystal structure you're looking at that location. We also have another wide field microscope. So this has a live cell chamber on it. It has three cameras, black and white color, and a SCMOS camera. We can set up perfusion for this, and then we have several boxes of cubes for this. So we can find the cubes you need for this. One of the projects that's been going on here is with the Han group and Alex. And he looks at, he does spatial genomics on, currently he's doing bee brains. And so this is a long-term experiment. You flow in a label, you image, you flow in another label, and then you image, and so on. And so you may notice that he uses quite a bit of time on this microscope. As a result of that, Duncan is building a home-built fluorescence microscope that's specially set up to do the serial fish type measurements like this. And so if you're interested in doing spatial genomics or serial fish imaging, Duncan or Alex are good people to talk to. We have a V16, so this is for larger scale samples, things like dishes that you want to see the whole dish for. So it has 1 micron resolution. It has fluorescence capability. It has a nice stage. It's not in this image here, but it has a nice XY stage for doing tiling. So there's a coal ball collection on campus, and so you can cut the coal balls, and so these are then have fossilized plants inside them. You do a peel off of that, and Sarangi's group has been using this to tile those very effectively. Another tool we have is an X-ray CT system. So there's X-ray CT tools up at Beckman at their microscopy core, and they're designed for high resolution imaging and small samples. This is designed for big samples, and so this image is of our system. It's actually the size of a small room. You can walk in there and set your sample in there. We can shoot X-rays through about 3/4 of an inch of steel, and so a lot of the samples that don't fit in Beckman systems can fit here. We can also do tiling, and so the detector moves around. So if you have a very big sample, like we were imaging a heat exchanger, which was like around this big the other day, and you can move the detector around to do tiling. So here are some of the kind of samples. On the small side, this is a leaf, and these are little air chambers going through the leaf. Here's the center rib of the leaf. Here's a fish from the Natural History Survey. We imaged, Andrew Leakey's group imaged a few hundred root crowns last fall. Here's an insect that's been stuck in amber for about 20,000 years. So there's all kinds of interesting things you can image with an X-ray CT system, right? We have a 600 megahertz NMR, and Ling Yang from the NMR facility in chemistry does the training on this, and so if you need trained or know someone who wants to be trained on the NMR in IGB, fill out the training request form, and I forward that to them, and then they set up training for that. And here's a project that Kingsley's working on. We have a serial block face scanning electron microscope also. And so imagine a tissue, like a millimeter by millimeter cube tissue that's been stained with heavy metals. And then Kingsley mounts it in our scanning electron microscope. We have a diamond knife that cuts the top off the sample, and then we image it, and you can come up with some kind of image that looks like this, where you can see the structure in there. And so all the cell walls, all the organelles, everything is labeled. And so this works automatically. So you can go section after section after section. So you can collect hundreds to maybe 1,000 sections. And Kingsley watches this overnight and just chunks away, right? So it's pretty effective at collecting sections maybe every minute or couple minutes. But then to get interesting information from this, you have to segment this image. So that means you draw around whatever organelle you're interested in, and if you do that through hundreds of sections, you end up with a really nice image that looks like this. So now you have information on the volume of some organelles in your tissue with about 20 nanometer resolution. So that's great, with one exception, which is that while we can collect data at maybe an image every couple minutes, it takes maybe up to an hour per image to do the segmentation, which gets kind of old and boring, right? So to solve that problem, Umina and Kingsley and I have started looking at different software projects or products to use artificial intelligence to identify and segment this data. And so our hope is to speed this up immensely. We've started a project where Umina segmented a set of data by hand, and I ran the data through a Zeiss product, and she's going to run the data through Amira, and we're going to compare the results and see how it works. So the hope is that this will speed up immensely. Once we get this data analysis sped up, then I think moving from the sample to usable information is going to be much, much easier. So following that, we have a couple other software products. One is AutoQuant. So AutoQuant does, what does AutoQuant do? AutoQuant takes images, it increases the resolution by modeling the point spread function. Amira can measure volumes of fluorescent samples. We can measure the areas and angles and widths and whatnot from nanozoomer images, and then occasionally we do things like plot the path of a fluorophore with from our data from our MinFlux. And so this is a fluorophore tied to a gel, and you could see there for a minute that it was bouncing around, and you could plot that path. Okay? So that is, whoops, so my image got too big and covered up our website. This is the last slide, and yeah, so are there questions? Yes.
How would the AI software you're involved with, is there any way to get involved with that?
Sure. What did you want to do with it?
Well, I'm a computer science student, so I thought it was an interesting project that I'd like to look at.
Come and talk to me afterward, because we would love somebody to do it. I really don't know that much about AI. So, yeah, that would be